Welcome to Nottingham British School, the home of science, maths and technology. I'm Dr. Zia Zamir. In this lesson, we're going to follow up from our enzymes definition to the functions of enzymes. We're going to talk about the substrate, the active site and the activation energy. So basically we understand what enzymes are. So we're now going to learn how these enzymes function to lower the activation energy of a chemical reaction. Enzymes bind to their substrates to perform all kinds of important and essential cellular processes, as well as processes that help you enjoy a slice of pizza. Okay, we'll start by enzymes. Lower the activation energy. I come from England, so basically we used to see a bit of snow, probably a couple of months of the year. So I'll use this analogy. Making a snowman is always a big ball of fun. 3 snowballs full of fun, to be exact. And each snowball starts out smaller, getting bigger as you roll it in white snow, basically. Let's imagine that you go outside in a fresh snowfall to bind one of these guys, but you don't have the best suited yard for rolling the snowballs. In fact, your backyard is one giant hill. This hill is fantastic for sledging, but not so much for making snowmen. Anyways, in order to make a giant snowball for your snowman's bottom, let's say, you'll need to actually push the snowball up over the hill and then down the other side. You do this because it's fun, but make no mistake, it takes a lot of energy to get started up this hill. This is hard work. Right now, you share a quintessential problem with some chemical reactions within the cell. These chemical reactions might be a lot of fun when you're a cell, but it takes a lot of energy to get them started. This is called activation energy. Okay? That's what's called activation energy, or the energy required for a reaction to start. You can think of activation energy as the energy you needed to muster up in order to push that snowball over the hill in your backyard in order to maximize the size of your snowball and get it to the other side. So, what your cells have to help overcome a problem of high activation energy is called enzymes. Enzymes are proteins that lower the activation energy of a reaction. In doing this, enzymes increase the rate of reaction, helping it to occur faster. However, enzymes are not consumed in a reaction. They simply help it to occur. They do not add snow to your snowball. They just make it easier to make the snowball. This makes enzyme reaction catalysts, the presence of enzymes to decrease the size of the hill in your backyard. Enzymes make things easier for your cell and help chemical reactions occur. There are hundreds of different kinds of enzymes in your cells, which all participate in different types of reactions. Enzymes can break molecules apart, build or add molecules, or even rearrange them. In lowering the activation energy of a reaction, enzymes decrease the barrier to starting a reaction. It is important to note, however, the change in energy remains the same between the start and the end of a chemical reaction. For example, if a reaction released is 200 kilojoules of energy without an enzyme, the same reaction would still release 200 kilojoules of energy with some enzymatic aid. The difference would only be a lower activation energy and a faster rate of reaction. In other words, if enzymes were helping you build a snowman, you'd be done quicker, but you'd just have just as much fun. Most of us are fortunate enough to be able to enjoy a grilled cheese sandwich, a hot chocolate when we're taking a break from building our snowman outdoors in the cold. This is because our cells contain an enzyme called lactase. It is interesting to note that remember the many enzymes names end in the suffix ase. Lactase breaks down lactose sugars in the glucose and galactose. Lactose, of course, is sugar found in dairy products. Some individuals are lactose intolerant and they lack this enzyme. Without lactase will break down on its own, but it is an extremely slow process, maybe even taking days. Lactase speeds up the rate of this reaction, allowing us to digest lactose. Let's talk about how it's done. Lactase starts its reaction by binding to its substrate. A substrate is a substance or molecule on which an enzyme functions. So the substrate for the lactase is lactose. Together with enzyme substrates form an enzyme substrate complex. So remember that word, enzyme substrate complex. Enzymes like lactase are block-like global proteins with pockets. These pockets contain the active site, which is the area of an enzyme when the substrate binds and the chemical reaction takes place. In the active site, amino acids of the enzyme protein will bind to the substrate. The substrate fits perfectly into the active site of an enzyme, meaning that enzymes are specific for their substrates and not for any others. Lactase cannot break down any other. For example, when binding to a substrate, enzymes may undergo an induced fit. This is the change in the enzyme shape to accommodate and bind to a substrate. Although it is true all substrates are specific for their substrates, you can imagine that this is just like a little tweak to an enzyme substrate complex to make the reaction occur. This change does not need to be drastic. It might be just like cradling a baby, then snuggling the baby into a position closer to your body for a better hold. Okay, let's look at this lesson summary. In summary, enzymes are proteins that lower the activation energy of a chemical reaction. Enzymes are not consumed or produced in a reaction, but they are catalysts that allow the reaction to proceed at a faster rate. There are hundreds of enzymes in each cell that has a specific role and work on a specific substrate. Together they form the enzyme substrate complex. Substrates bind to a perfectly matched pocket in the enzyme known as an active site.